Guys, so today we're going to talk about what must happen before a cell can divide. And we talked about this in our mitosis and meiosis unit. So before it can do that, we have to check to make sure everything's good. But the big thing that happens is DNA is synthesized, and that's in that S phase of interphase. And so how that happens is what we're going to talk about today. And so we're starting with one strand of DNA, and we're going to end up with two strands at the end. Okay, so there's steps of this, and I don't really care that you know like and memorize it step by step, but just kind of knowing the overall process of how that happens. So our first steps kind of happen at the same time anyway. First thing that's happening is the DNA is unwinding. So here we have the DNA all wound up, and you can see that it is unwinding. So that means the double helix unwinds, and a protein is going to be doing that here, shown in blue. Also, it is going to be unzipped. So that's these hydrogen bonds that are along here are going to be broken, that are connecting those base pairs together. And it makes this kind of Y shape that we can see here. So these kind of happen at the same time. So the big idea is that it's untwisting and unzipping the DNA. The next thing that has to happen is primers have to be added. And this is kind of a confusing part and a thing that's helpful to know about the difference between DNA and RNA. DNA cannot be synthesized what we call de novo, from nothing. So you cannot just start making DNA molecules, or our enzymes are not able to just start making DNA molecules and synthesizing them by adding bases from nothing. But RNA actually can, and this is all dealing with that OH group found on RNA that's not found on that deoxyribonucleic acid. So first what happens is a short RNA primer is added to the 5' end of the molecule. And notice, remember, DNA is antiparallel. So here the DNA is going 3' to 5'. It's going from the 5' end to the 3' end. So an enzyme comes in and it starts adding these bases in, but these are RNA bases. Same thing will happen on the other end. The next thing that happens is we have DNA that is replicated, and this is complementary to. So since there's already RNA here, DNA can just hook onto that, and it can start synthesizing now by adding new bases into the spot after this RNA primer. And again, notice we're going from 5' to the 3' direction. Same thing here. Okay, and then the next thing for DNA replication is we need to remove those primers made out of RNA. So we're going to remove the primers, and the backbone is connected where the two DNA molecules were. So the enzymes that do this are important. We have topoisomerase, that's here now in green, and it was blue in my picture before. That's the enzyme that untwists the DNA here. We have DNA helicase. This is the enzyme here in this blue triangle. That's the enzyme that is breaking the hydrogen bonds. We have single-strand binding proteins. Here, these are shown as these purple ovals. Those are the things that just help the DNA from folding back in on itself to bond back, because these bases want to hydrogen bond back together, so they kind of get in the way to prevent it from just bonding back together. We have primase, and as its name suggests, it's what adds the primer, the RNA primer. So it's adding RNA from that 5' to 3' direction. We have DNA polymerase 3, and there are different DNA polymerases. We have 1, 2, and 3. They all have different jobs, but really getting into how they're different isn't super important for this course. But DNA polymerase 3, it's going to be adding those new DNA nucleotides on, 5' to 3', adding on from that RNA primer. DNA polymerase 1 up here is what removes the RNA and then in its place adds DNA. And then ligase is important over here in blue because it's going to actually bond those two DNA segments together where that RNA was replaced and that DNA had been synthesized. So it just bonds those two, the backbone, together. We do have DNA polymerase 2, and it goes in through and repairs errors in the sequence, but it doesn't have like a main role in the actual synthesizing of the DNA other than checking for errors.